Everyone, I'm Ryan Drum. I'm a conservation biologist with the U.S. Fish and Wildlife Service, and today I'm joined by a national team of biologists from across our agency, from across the country, representing a variety of perspectives and the many ways we're engaged with native bee monitoring efforts across Fish and Wildlife Service. We're going to focus in on how and where we're collecting bee data. We'll look at those areas where we can contribute to the bigger picture of bee monitoring activities. We'll also discuss how we're using these data to inform habitat conservation efforts and regulatory decision making. It's really an exciting time for bee conservation and pollinator conservation more broadly. There's just a sense of growing awareness, interest, growing urgency, you could say, from across our agency and beyond, and across our leadership as we look to understand what's really happening to bees and what can we do about it. So we're really happy to be here today to share, to learn from others as we explore how to best align our efforts. Today we have 6 speakers, including myself. Each will offer a glimpse into the different ways we're engaged in the native bee monitoring arena. I lead our national level pollinator science coordination efforts. Lee will speak to inventory and monitoring across the National Wildlife Refuge System. Tam is going to focus in on Endangered Species Act regulatory aspects. Mary will describe bee monitoring activities that are occurring in the Great Lakes region. And Claire is going to describe how we're building capacity alongside USGS to identify bees collected in the field. And lastly, James will close us out with geospatial tools that are helping us to prioritize where we focus our conservation efforts. The mission of the U.S. Fish and Wildlife Service is to work with others to conserve, protect, and enhance fish, wildlife, and plants and their habitats for the continuing benefit of the American people. You might think about this as having 3 elements. We focus on the fish, wildlife, and plants, so the organisms, their populations, and also the habitats upon which they depend. We do this working through a whole variety of collaborative partnerships and ultimately to benefit the American people. Fish and Wildlife Service is a bureau within the Department of Interior, which means we sit alongside a number of other agencies you're likely familiar with and some of whom you'll be hearing from as part of the workshop series today. The scope of our mission is quite broad. There's many layers to our organization, many programs through which we might connect to bee monitoring efforts. Our work spans from public and private lands management to regulatory decisions. We operate regionally, nationally, and internationally. So there are immense opportunities across our programs to support and use native bee monitoring data. But a big part of the challenge we face, frankly, is tracking everything we're doing, standardizing efforts across these various dimensions of our mission areas. In a sense, we're only just beginning to truly get organized around bee monitoring efforts nationally. And so this is a really timely conversation to drive a more coordinated and strategic approach. A big part of what we have to offer centers around our public lands that we manage and monitor. So I'll go ahead and pass it over to Lee now to share more about the status of our efforts across the National Wildlife Refuge System. Go ahead, Lee.
Hi, my name is Lee O'Brien. I'm an ecologist with the Inventory and Monitoring branch of the National Wildlife Refuge System. And I'm going to talk a little bit about what we're doing to support bee monitoring on wildlife refuges. One of the things we've done is gather together bee monitoring experts and put together this survey protocol. The protocol covers several different methods of bee monitoring, from bee bowls to vane traps to netting and photography. And there's sections on data management, vegetation sampling, and suggestions for how to get your bees identified. We also have this electronic data collection tool that you can put on a laptop and enter data from bee surveys. It was tailored to wildlife refuges, but anyone can use it and put their location in the tool. And it's specific to different types of survey methods. So you select from the drop-down box which method you're using, and a different page will open to specific to that method of survey. Once you select a method, this page will open, and the top part is the same for all surveys, as the date, conditions, ecosystem type, and the bottom part is specific to the survey type. There's also a button on here to make labels, which will generate unique labels for each specimen, and based on the location and date. And you can attach those labels to your specimens and send them off to get ID'd, and then once they're ID'd, enter the identification in the tool. The protocol, database tool, paper data sheets, and how-to documents can all be found on our documents catalog called ServCat at the URL there at the bottom, or you can contact me and I can send you the link to this page. We also have a database of what type of surveys are being done on refuges. It includes the name of the survey, the species that are being surveyed, what region, the refuge name, the status of the survey, and the type of survey. As you can see at the bottom there, as of June 23rd, there were 67 historic bee surveys on refuges, 80 current bee surveys, 2 of them expected to be done this year, and 6 expected to be done in the future. We also put together a page for bees and wasps observed on wildlife refuges on iNaturalist. So refuge staff can upload their photographs of bees here, and folks will come in and identify them. And if you get a research grade identification, you can enter that ID into the data collection tool. And citizen scientists can also use this page for observations of bees and wasps on wildlife refuges. And then it collates them by refuge, and so then we can go in and see what bees have been observed on each refuge. We also have help documents in ServCat that tell you how to set up an account on iNaturalist and how to enter data. So in conclusion, I'd like to mention a few things that we at Refuges would like to see in a national pollinator monitoring program. One is national sampling design, so that we know which refuges could contribute to national status and trends of bees. We'd also like to see a national database where we could upload our data. And finally, more capacity and a better system for getting bees ID'd, which you'll hear about a little later in the presentation.
Hi everybody, I'm Tam Smith with Ecological Services in Minneapolis. Ecological Services is the Fish and Wildlife Service program that administers the Endangered Species Act, working with experts in the scientific community to identify species on the verge of extinction and to build the road to recovery. The types of bees that we monitor and survey focuses on threatened and endangered species, species under review for potential listing, their habitats, and associated species. The data we collect is used to inform species status assessments, listing decisions, recovery decisions, Section 7 consultations, 5-year reviews, post-delisting monitoring, and to improve protocols or to assess threats. If you're interested in the details of our status assessment and listing process, please watch the Fish and Wildlife Service talk from the October workshop. While Ecological Services often relies on external data collection, there is some internal monitoring that we do. Because we focus on specific species, though, our methods and protocols will differ between them. However, most of our monitoring includes some common metrics, at least, presence, location, habitat metrics, and presence of threats. The exact protocols used may vary by taxa, and I'll provide a few examples of those on the next few slides. For example, recent Bombas Franklin surveys are conducted primarily by Fish and Wildlife Service with help from partnering agencies. They use a protocol developed by the Western Bumblebee Advisory Group that largely follows Robin Thorpe's survey protocols with added statistical rigor. And they use time surveys in a set area and record Bombas community and their floral associations. They store the data at the Fish and Wildlife Service office and share their data with the Pacific Northwest Bumblebee Atlas as well as others. While Fish and Wildlife Service does conduct some Bombas affinis surveys, the vast majority of our data come from permit holders, state and university programs, and community science data sources. The affinis protocols were developed to provide a variety of options for non-lethal surveys to satisfy multiple objectives that other entities may have, but still get us some of the basic information that we need for tracking recovery. They range from photo-only quick site surveys to rigorous timed area or distance sampling with a minimum time per unit area with 4 visits per site per season. We're currently working with USGS to improve our more rigorous protocol to include measures of detectability and refine needed effort to be confident in our results. We track recovery based on a set 10 by 10 kilometer grids and have online maps of grids where we encourage our partners to survey. All surveys ask for associated Bombas community data, floral associations, basic habitat information, and we also allow permit qualified individuals to conduct non-lethal sampling of genetics, pathogens, pollen, and to collect data at nests. We have developed a standardized form that permit holders are now required to use, and we store data at one of our field offices, not a centralized database for external use, but we do provide maps for the public to use. The Hawaiian yellowface bees are surveyed primarily by university partners, and because of the difficulty in sampling, the surveys are presence-absence. They don't quantify abundance. The searches focus on the areas of suitable floral hosts within suitable habitat during favorable weather conditions and during the peak summer flowering season. The capture rates are low and they're difficult to detect. In addition to habitat data, they document the potential competitors or predators. Bombas occidentalis monitoring is conducted primarily by U.S. Fish and Wildlife Service and partnering agencies. They use the same protocol developed by the Western Bumblebee Advisory Group that I described for Bombas franklini, and they've identified areas where there's knowledge gaps for the species and encourage surveys in those areas. The objectives are to estimate spatially explicit trends, evaluate the range extent, and estimate current occupancy. They use time surveys in a set area and record habitat and floral associations. The Fish and Wildlife Service folks working on assessing the blue calamintha bee largely rely on surveys conducted by the Jarrett Daniels Lab from the Florida Museum of Natural History to inform the SSA in the 12-month finding. They have 4 major objectives. The first is to determine the status of the bee in the floral host, where the methods they use focus on areas with ash calamint. They select those areas with the high densities of that plant, meander, and then sit stationary until the bee is observed. They have other objectives to understand natural history and habitat, evaluate future distributions, and develop monitoring and habitat management recommendations. And they also collect pollen samples to determine which plants the species visits and take DNA samples to determine how genetically isolated populations have become. There are some restrictions for collecting and sharing data. For example, recovery permits may be required if you think you may handle a listed species. And data sharing can be restricted as data sharing agreements vary from state to state. Also, some data is considered proprietary, which may complicate data sharing. As for data management, Ecological Services does not typically use a centralized database and typically store data at individual offices, although they may share between offices. In summary, while we do some monitoring internally, Ecological Services often relies on data provided by external partners. We tend to focus on areas where we know the species is to be or where we expect it to be, although sometimes we focus on areas where we know that there's little effort. To inform future SSAs, recovery plans, and other ES needs, it would be beneficial to collect additional data, such as specific impacts of stressors, life history information, and habitat associations, as well as demographics and health metrics. I think also Ecological Services would benefit from having a centralized, standardized, and accessible database with enough detail to track species recovery, for example. Thanks, and I'll now turn it over to Mary.
Hi everyone, I'm Mary Holm with the Services Partners for Fish and Wildlife program. In the Great Lakes region, the Partners program is part of the National Wildlife Refuge System. With funding from the Great Lakes Restoration Initiative, on behalf of the service, I am coordinating a multi-agency GLRI pollinator task force. The task force is focused on restoring habitat for native bees. But in order to make critical and informed decisions about where and how to restore habitat for native bees, the task force agencies are conducting research and native bee surveys. Agencies involved in the task force include the Fish and Wildlife Service, National Park Service, U.S. Forest Service, U.S. Geological Survey, Natural Resource Conservation Service, and Bureau of Indian Affairs. Partners conducting native bee surveys within the task force include the Fish and Wildlife Service, National Park Service, U.S. Geological Survey, and the U.S. Forest Service. In order to inform habitat restoration and management decisions on lands of these federal agencies, one of our first steps is to find out what we have, what's present on our lands. So sampling native bees is an important part of task force efforts. One of the task force's goals is to work together to develop a common standardized protocol that all of the task force agencies could use to gather comparable data on native bees. The Pollinator Task Force survey team has been working on this task for about the last year. The team did a comparison of protocols and found some common themes. We are all using bowls, netting, and are gathering some kind of vegetation data during our surveys. Though we are all using similar protocols, there are differences in the details. For example, the length of the transect, bowl placement and number, whether the transects are linear or circular, the frequency of sampling events, and the type of vegetation data collected all differ slightly. Most of the survey team's time has been spent on nailing down our objectives as a group. Here you see basically a list of everything we could think of. We're still working on prioritizing and refining them. We need to know what we have and inventory, but we would like to gather information to inform other objectives as well, like relative abundance, how management affects species, species assemblages, and more. The main reporting metric of the GLRI Pollinator Task Force is habitat acres. With that in mind, the team has most recently determined that a sampling goal should tie directly to habitat and habitat management. We would like to provide information to land managers to help them make future decisions. We want to collect data to help them understand risk, make the right decisions for current and future species, understand the relationship between bees and available habitat, and protect and improve rare habitats and/or rare species. We want information on relative abundance and trends in species and communities, including before and after management actions. We are working on prioritizing surveys based on these specific goals. Some of our most recent discussions revolve around focusing on rare species and rare habitats, with each agency targeting and surveying specific habitat types. The team has also discussed how we could potentially play a role in helping to refine existing sampling techniques to learn how better to detect species that may not be currently detected with existing techniques. There is also a desire to get away from destructive or lethal sampling and overly laborious identification. The Park Service, U.S. Geological Survey, and Forest Service are testing out eDNA collection techniques for bee identification and plant associations. Specific to the Fish and Wildlife Service efforts within the task force, 2020 was our pilot year of bee sampling at Shiawassee National Wildlife Refuge and a couple waterfowl production areas in Michigan. This year, we sampled 5 refuges and 4 waterfowl production areas in Ohio, Michigan, and Wisconsin, as well as 1 private land site in Michigan. We had a total of 130 sampling events, visiting each site approximately once per month, May through October. We based our site-specific sampling protocol on the national framework using bee bowls and targeted and opportunistic netting. We recorded the flowering plants at the site and the plants on which we collected bees during each visit. We used a simplified data sheet seen here and entered all data collected into an Excel spreadsheet stored on the computer in our office. Some other options we are considering for data collection in the future include using Survey 123 or the Collector app on mobile phones or tablets, and diving into some eDNA work ourselves, collecting pollen during our surveys. Next year, we will expand our native bee sampling to Region 5 of the Fish and Wildlife Service. This will include 2 refuges in New York, Iroquois and Montezuma National Wildlife Refuges. We are also doing survey and bee identification work with partners. We are working with Michigan Natural Features Inventory on a project that includes surveying bee communities of rare habitats, including Lake Plain Prairie and Prairie Fen, conducting a conservation status assessment of megachile, and conducting bomba surveys at Michigan National Wildlife Refuges. We're also doing work on bee identification with partners. Our 2020 bees are headed to the University of Minnesota for identification by Zach Portman at the Caribou Bee Lab. And our 2021 bees will be headed to Patuxent for identification by Claire Maffei of the service and Sam Droege of USGS. As far as data management goes, besides our Excel spreadsheets with the metadata, we use Discover Life to print our labels, and once the bees are identified, that data will be entered into Discover Life as well. A main goal of the task force is to find a database that all partners can use and agree upon to store and share data. The Fish and Wildlife Service and the Pollinator Task Force will continue to work with regional and national partners to assess goals and objectives, develop a sampling protocol or set of them, and test new methods of sampling and identification with the hopes of providing data that will inform land managers and others and help them conserve native bees and their habitats. Thank you for your time today.
Hi friends, welcome to the USGS, U.S. Fish and Wildlife Service Native Bee Lab. I'm Claire Maffei and I am with Fish and Wildlife Service Science Applications Headquarters. So we're a support lab, and what we do is provide, among a lot of things, is bee identification for our agencies and the many other groups who do field sampling. As you can see, and are probably already more than well aware, there is a significant bottleneck between projects collecting bees and the people available to identify the specimens. Our lab is one of the places that gets to work on those bees all the time. But we still want to address this skill gap. So beyond doing the identifications ourselves, we're building out training programs to support workforce capacity. For example, on Wednesdays, Sam Droege and I use this wonderful microscope and camera setup to offer a live streaming bee identification course where we walk through bees underneath the microscope. By translating jargon into terms and characteristics that more biologists can access, we're hoping to eliminate the silos around this tricky skill set. Mary and the GLRI team will be sending us their bees next year. Paul Charland, a service biologist in that group, recently detailed with us, and so he's able to organize the bees to genus level before they come our way. We're really excited about investing in a model that builds regional expertise, and it allows us to turn around data that much faster. We all know this will be especially critical as the bee community continues to scale up our surveys. So we invite you to come out and learn your bees, and hope to see you soon.
Hello RCN, I'm James Weaver in the Science Applications Department based in the Southwest Regional Office in Albuquerque, New Mexico. I'm currently developing fully interactive decision support tools focusing on known bee distributions across time using publicly available occurrence records provided by GBIF, shown here on the left. The tool incorporates NatureServe conservation status rankings and allows users to determine apparent bee richness across varying spatial and temporal scales, and can highlight geographic areas which may have high concentrations of at-risk bee species diversity. The tool can also highlight areas which are relatively depauperate in historic sampling, potentially allowing us to focus on areas which are in need of additional monitoring efforts. Pages 2 and 3 of the tool also includes bee occurrence records across Canada and Mexico. More recently, we have added the capacity for the tool to assess bee monitoring efforts from within Fish and Wildlife refuge properties across the United States, as seen here on the right. This will allow us to catalog bee taxa within each refuge, highlight refuges with little to no historic monitoring, and track changes in bee community composition over time. Data from future monitoring efforts, such as from individual refuges explained by Lee O'Brien, can be easily incorporated into the national database in order to have a central location in which to visualize results from bee monitoring efforts from within the service nationwide. We are currently in the process of expanding this functionality to other federal land management agencies such as the Bureau of Land Management, the Park Service, Forest Service, and Defense Properties. In summary, our efforts nationally to prioritize wild bee and pollinator inventory and monitoring and conservation in a more broad, holistic approach is still in its relative infancy. Objectives, protocols, and sampling efforts and designs vary among our internal programs. But our overall goal remains the conservation of species, ecosystems, and the land. As explained by our previous speakers, we are currently deploying some of these conservation efforts at local and regional scales, and we hope to expand these efforts more widely in the coming months and years. The service has a team of talented biologists across an array of departments, such as the National Wildlife Refuge System, our regulatory, science and training and bee identification teams, who are passionate about native bees and pollinators as a whole. And while we still have a ways to go, we are committed to working with our other federal land management partners, the academic community, and private and nonprofit partners to provide meaningful and effective inventory and monitoring efforts across our own managed lands so we can better understand population trends and initiate conservation delivery measures moving forward. Thank you, and we hope you enjoy the remainder of the presentations.